Alright, to wrap up the few remaining topics of this unit on the brain, I'd like to talk for just a minute about baseball. Last year, 2018, I believe it was the Dodgers that won the World Series. I want you to consider for just a second what it takes to actually hit a baseball. Let's orient ourselves. We've got some internal structures in the brain displayed below. Of course, we would draw an eyeball out here. That's going to be connected to the cranial nerve. It's going to come through here. Think about what this signal, this is an afferent signal coming in through cranial nerve 2, the optic nerve. Think about where this goes. We already have determined that all of the sensory is first going to come in right in here to the thalamus. In the thalamus, except for smell, in the thalamus, the sensory is sorted out and parsed into certain areas in the brain. One of the areas where this is going to go certainly is going to be back here. The occipital lobe is the area where vision is processed so that we can actually see a map and the brain can interpret that map accordingly. There's a lot of sorting that goes on in the thalamus before it gets there. One other area where this is going to go is down here to the superior colliculi. We listed these in a previous lecture, described them as the corpora quadrigemina, the superior and inferior colliculi. We failed to mention what they were for. These structures are to assist head movements in response to either sight or sound. The superior colliculi will track the head based on sight. The inferior colliculi will track the head based on sound. Consider for a second somebody blows up a balloon and starts tossing it around a room. The ability for you to see the balloon, watch it move, and track your head accordingly is part of the job of the superior colliculi. And so in this sense, we've got cues coming in from the eye as the pitch comes, and the eye quickly relays information to these certain areas of the brain. The superior colliculi will allow the head to track the ball, and then patterns that we see in how the ball is moving and how the pitcher releases the ball are going to help the brain determine whether it is a fastball or a slider or a sinker, all of these different pitches that might come. And then from that information, you're going to execute a motor judgment based similarly on what we talked about before. Is it a good ball to swing at? Is it a bad ball to swing at? Let's weigh the checks and balances and determine what we want to do. So once a motor program is determined, then the thalamus will send its signal out to the motor cortex. The motor cortex, the premotor cortex is involved in planning. That's going to relay information to the primary motor cortex. And then we've got, from the motor cortex, action potentials coming down into the brainstem, into the spinal cord, that are going to synapse on lower motor neurons to initiate a response. However, one thing that's really important to recognize is that these batters batting in the World Series or any Major League Baseball game have swung the bat thousands and thousands of times. And the swing is polished, and it's not clunky, and there's detail in every movement. And that's a type of memory that I would like to talk about for just a second. So let's consider for just a moment the concept of memory. If we define learning simply as the acquisition of knowledge, then memory simply becomes retention and storage of that information. What I would like to do now is break this down into two basic components. We can break this down into what's called implicit memory and explicit memory. We've already discussed some aspects of explicit memory. This is, of course, going to be declarative, and it's going to involve the hippocampus, which is specifically involved in declarative and short-term memory. And this is based, like we said, declarative. It's based on remembering events, words, rules, and language. We could even further explore short-term memory. There's still some debate as to what exactly is short-term memory, but one thing that I would like you to recognize, working memory is very short and is limited to 7 or 8 bits of information at a given time. I can transition that working memory into short-term memory and then long-term memory simply through repetition. It helps if I can make associations. And of course, emotion plays an important role in the formation of that memory as well. Now let's go back and let's look at implicit memory. This is non-declarative and what we would call reflexive. This is going to include procedural memory that can be broken down essentially into skills and habits. And the thing that I want to make sure you understand is that for implicit memory, the hippocampus is not involved. So if the hippocampus in the limbic system is not involved, where does this implicit memory come from? Where do we store our muscle memory? If we go back to our picture, it's certain that the thalamus and the basal nuclei or the basal ganglia that we previously discussed hold some aspects of balance and posture and executing motor programs. But the fine-tuning of those motor programs, what gives us that wonderful muscle memory, is a structure back here called the cerebellum. As upper motor neurons come from the motor cortex down through the spinal cord, the cerebellum is in a perfect location to go in and through synapses, through interneurons, through all sorts of different connections, provide support and guidance to fine-tune that muscle coordination. Try and think of the first time you threw a baseball. Think about how awkward it was. Or maybe if you can't remember that and you watch somebody who hasn't ever thrown a ball, watch them try and throw a ball, and it seems awkward and it's hard to watch. But over time, you build a muscle memory. You start to recognize, I should do this, I shouldn't do that. And as your implicit memory builds upon those experiences, you're able to develop a pattern that allows you with precision to execute the fine-tuning of that motor program. That's the idea behind implicit memory. Well, there's some who could say, I could hit a baseball in my sleep. Well, that might actually not be too far from the truth. Let's consider what it takes to be actively engaged in what we would consider deep thought or deep learning thought. These types of studies, as we look at brain waves, have their roots in the use of an EEG or an electroencephalogram that identifies patterns of neuron behavior found in the cerebral cortex. They're specifically focusing on cells found in the cortex called pyramidal neurons. These pyramidal neurons have triangular soma, as we've drawn, and they're a little bit unique in that. And what the machine is able to do is detect a dipole. This may seem like a foreign word to you, but a dipole is merely separation of charge. And whenever I separate charge, I create an electrical field that can be picked up by a sensitive machine. So if you consider how neurons work, if we have a lot of input, say an EPSP, that comes into the dendrites of this cell, we would expect to see a lot of positives build up on this end of the cell. We know that those positives are going to travel down to the axon, where if there are sufficient positives, we could elicit an action potential as threshold is reached by voltage-gated channels beginning at the axon hillock. But in the meantime, this area remains negatively charged. And of course, we're talking about what's inside the cell. It would be just the opposite for what's outside, what the machine, the EEG, is actually reading. What I want you to recognize here is that with this separation of charge, a lot of positives on one end and negatives on the other end we create a dipole. And the frequency of dipoles allows us then to measure the frequency of brain waves. We make this measurement in hertz, or Hz, which is 1 wave per second. We can use that then to look at different brain waves available or observed by this machine. We have alpha waves, beta waves, theta waves, delta waves, and a fairly recent discovery, gamma waves. These are all characterized based on the frequency, or how fast these neurons, these pyramidal neurons, are firing. If we look at alpha, these tend to be anywhere from 7 to 14 hertz. Beta is higher, from 14 to 30 hertz. Theta is going to be lower at 4 to 7. Delta even lower than that. We're going to say less than 4. And gamma, we're going to say higher than 30. So what does this mean? Well, based on these patterns, we can use this to predict the level of consciousness or awareness that brain is experiencing based on its firing rate. So with alpha, this tends to be awake, but in a very restful state. If we contrast that with beta, we see higher frequency of waves. That's going to suggest concentration and study. Theta, on the other hand, is going in the opposite direction, and we'll see this in a minute. This is entering sleep patterns. Delta is the lowest level of consciousness that we see. We see this in heavy sleep. Gamma had been predicted for a long time, but with analog EEG machines, it was not something that we could really measure. However, with new digital machines over the last several decades, we've been able to verify that these are here. This is what we would consider enlightenment, a high level of processing. This doesn't happen every day. For some people, it probably never happens. Buddhist monks have shown that compassion meditation allows them access to gamma waves on a regular basis. So there are things that you can do to train your brain to reach this state of enlightenment. But for most of us, throughout most of our days, we're not going to see consistently those types of waves. Now, how might this be related to sleep? Well, if we look at sleep, and I'm the first to state that this is a little bit ambiguous, and you can look in different textbooks and find lots of different explanations. But for now, we're just going to keep it simple. We're going to follow sleep as a simple process from non-rapid eye movement, or Nrem, and we're going to have several stages of this: Nrem 1 to Nrem 2, to Nrem 3, to Nrem 4, followed by REM sleep, or rapid eye movement, and that's going to come back and start the process again. The first time, maybe about 90 minutes, increasing in time throughout the night. So it becomes shorter and shorter. Now let's look at the stages. In Nrem 1, what would we expect? We would see alpha waves with easy arousal possible. As we transition to stage 2, this is going to be characterized by mixed waves, and it's going to be a little bit harder to arouse. By the time we hit stage 3, we've got predominantly theta waves, some delta, and here we see the vital signs decline. And then the last phase of non-REM sleep, predominantly delta waves, lowest vital signs. This is where we see sleepwalking and also night terrors. From here, then we transfer into REM sleep. This brings us back to alpha waves. Our vital signs come back up, and we become very active, but this is only visible in our eyes because the reticular activating system in the brainstem paralyzes your muscles. So we've got paralysis that's stepped in to prevent events like sleepwalking. So back to that baseball swing. Certainly, if somebody was throwing a 95-mile-an-hour fastball at me, I would not be in anything but beta wave configuration as I try and focus on that ball and make sure that if it's coming at me, I'm able to move out of the way. But people who have developed patterns of behavior, the coordinated muscle memory, it becomes easy for them. And because in the early stages of sleep you have alpha waves that's also awake but in a relaxed state, maybe it is just possible, as long as you can see the ball, to hit that ball in your sleep.